Hello everyone, Mrs. Klein's here with your first section of video notes for this unit over the history and structure of DNA. Before talking about the structure of DNA, it is important to understand how far we have come in the understanding of the DNA molecule. The scientists listed on your screen have all made a large impact on the understanding of DNA. They paved the way for each other to continue further research, as well as enabled the future research to continue by modern-day scientists such as Craig Venter and Francis Collins. All of these scientists have changed not only our understanding of DNA, but our understanding of how to treat diseases, how genetic material is passed from one generation to the next, and how closely related we are to other species on our planet. So let's briefly begin by discussing these scientists. Gregor Mendel was an Austrian monk who is most famous for his experiments on pea plants and being called the father of genetics. Mendel's research discovered that you receive one of each copy of your gene from your parents, and depending on how the gene is expressed, will determine the physical appearance of the gene, such as traits being dominant or recessive. Friedrich Miescher was a Swiss physician who focused his efforts on research instead of treating patients. At the time, many scientists thought proteins were the inherited material, including Miescher himself. However, in his research, Miescher was able to isolate something called nuclein from cells in pus of used hospital bandages. Miescher determined that this isolated nuclein contained hydrogen, oxygen, nitrogen, and phosphorus. The nuclein material he had discovered was in fact DNA. He just did not know it at the time. Frederick Griffith was a bacteriologist studying the pneumonia bacteria using mice. At the time of his discovery, he was actually trying to create a vaccine for pneumonia. Instead, he found that DNA was the genetic material and not protein. Well, he actually did not realize that it was DNA. He just called it the transforming principle instead. As you see from the diagram on your screen, Griffith's experiments worked like this. He used two related strains of pneumonia bacteria. One was called R because it had a rough appearance and it did not cause the mice to become infected. The second was called S because it was smooth. Also, the S strain caused the mice to be infected and suffer from pneumonia. As Griffith continued with his experiment, he then heat shocked the S strain of the bacteria. This resulted in the S strain not infecting the mice as it had before because the bacteria had died. But the experiment became really interesting when Griffith mixed the R strain with the heat-shocked S strain. The mice that were injected with the mixed strains of pneumonia actually got sick and died. Griffith even found via blood samples that the bacteria in the dead mice contained pieces of the S strain bacteria that had been taken up by the R strain bacteria. Interestingly enough, bacteria will steal properties of or genetic material they find advantageous from other bacteria. This is how the R strain got the characteristics of the dead S strain and caused the mice to get sick and die. This is why Griffith called it the transforming principle. Like all good scientists, Oswald Avery and his peers piggybacked off of Griffith's work. Avery and his team set out to determine what exactly Griffith's transforming principle was, because at the time they did not know it was DNA. Avery continued to test Griffith's experiments by running further tests, such as investigating which elements made up the so-called transforming principle and doing chemical tests for the presence of proteins, which was considered to be the current inherited genetic material at the time. Erwin Chargaff made a great discovery in regards to the rules of base pairing, which we talk about on another slide. However, it is important to note that his discovery guided the discoveries made by Wilkins and Franklin, as well as Watson and Crick. Rosalind Franklin and her partner, Maurice Wilkins, are credited with discovering the basic dimensions of DNA strands from high-resolution photos of crystallized DNA fibers using X-ray crystallography. They also determined that phosphates made up the outside of the structure of the DNA molecule. Around the same time, and once again piggybacking off of the research done many years earlier, Martha Chase and Alfred Hershey determined that DNA was in fact the material that was inherited and not protein. They confirmed that DNA was in fact the transforming principle and therefore genetic material, which disproved previous ideas that protein was the inherited material. From the diagram provided on your screen, you can see that Hershey and Chase's experiment centered around bacteriophages, specifically viruses that infect bacteria. What they believed would happen was that the material injected by the bacteriophage into the bacteria was a protein. So they tagged one sample with radioactive sulfur, knowing that proteins contain sulfur and DNA does not. They also tagged another sample with radioactive phosphorus, knowing that DNA contains phosphorus and protein does not. After their experiment, they tested the bacteria samples and found only the presence of phosphorus, which meant that DNA was the genetic material, ruling out proteins. Researching at the same time, and really just down the road at another university from Franklin and Wilkins, were James Watson and Francis Crick. Watson is actually still alive today. Anyways, using what looked like a kid's playset, Watson and Crick built the first accurate model of DNA, essentially discovering the structure of DNA. Their discovery was based on the findings of Erwin Chargaff and also the contributions of Maurice Wilkins and Rosalind Franklin. Although there's a lot of controversy surrounding how Watson and Crick obtained the missing piece of their research from Rosalind Franklin. Moving into the 21st century, Craig Venter and Francis Collins, as well as many other scientists all over the world, are credited with sequencing the entire human genome, meaning they know every piece that makes up human DNA, all 3.2 billion base pairs. This has opened up further research into personalized medicine, how we treat certain cancers and diseases, and even how you are just prescribed everyday medication. Now that we understand how the information involving DNA all got here, let's reflect back really quick to our biochemistry unit. If you remember, there are 4 macromolecules and nucleic acids are one of them. Examples of nucleic acid polymers, meaning the large molecules, are DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Every single nucleic acid, whether it be DNA or RNA, is made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. We can thank Miescher, Wilkins, Franklin, Hershey, and Chase for that. The monomer or building block of DNA is a nucleotide, which you can see on your screen. Therefore, every strand of DNA or RNA is made up of many nucleotides. Now let's discuss the structure of DNA. If you remember, DNA is found in the nucleus of all eukaryotes, and the only time DNA does not exist in the nucleus is during cell division. All that being said, organisms like bacteria do not have a nucleus because they are prokaryotes. Therefore, their genetic material, DNA, floats around in a specific area called a nucleoid region inside of a prokaryotic cell. DNA is double-stranded, meaning one pair of nucleotides are on either side, which you can see from the middle diagram. It is the arrangement of the nucleotides and the fact that each individual strand of DNA runs opposite or anti-parallel of each other, which gives it its shape like a helix, almost like a winding staircase. As you can see from the image, the nucleotides or building blocks of DNA have 3 important parts. The parts include a 5-carbon sugar, a phosphate group, and one of 4 possible nitrogen bases. The nitrogen bases are adenine, cytosine, guanine, and thymine, abbreviated with A, C, G, and T. And these make up the middle rungs of the DNA molecule, as you see here. The sugar and phosphate group of each nucleotide build to create a backbone or spine of DNA, as seen here. You can also note in this image that the cytosine and guanine are held together by 3 hydrogen bonds, whereas the adenine and thymine are only held together by 2. Next, let's briefly discuss the function of DNA. This was previously noted when we were talking about the discovery and research that led to our understanding of DNA. First, DNA stores information. DNA is kind of like a giant recipe book in the cell that holds all of the instructions for building proteins. It also copies information. So before a cell can divide, DNA replicates or copies so that each new cell gets a complete set of this genetic information. And the third function is that it transmits information. DNA is passed on to new cells during cell division and helps to carry genes from parent to offspring. In speaking of nitrogen bases or rungs on the ladder in the previous slide, it is important to note there are two types of bases, purines and pyrimidines. Purines, like adenine and guanine, which you can see from the image on your screen, are larger because they are composed of two carbon-nitrogen rings, whereas pyrimidines, like thymine and cytosine, are smaller because they are composed of only one carbon ring. Thanks to the research of Erwin Chargaff, we now have Chargaff's rule. We know that adenine, or A, always pairs with thymine, or T, and cytosine, or C, always pairs with guanine, or G. Therefore, as you can see in the example on your screen, we know that whatever the amount of adenine is in an organism, there must be the same amount of thymine, and the same goes for cytosine and guanine. Now, an important note, if you noticed, a purine, like adenine, always bonds with a pyrimidine, like thymine. This is what builds the helix structure that DNA is so famous for. Also, the bond that holds each nucleotide to its complement is known as a hydrogen bond. There's a double hydrogen bond between A and T, and a triple hydrogen bond between C and G. This is why DNA that has more C's and G's can take more time and energy to separate. Remember, hydrogen bonds are seen everywhere, even from one water molecule to the next. Moving on to section 2 of your video notes over DNA replication. Before we dive into DNA replication, I wanted to talk a little bit about the cell cycle. This image here depicts the life cycle of a cell and different important things that happen along the way. You can imagine that the birth of a cell happens right here in G1. The cell spends most of its life in this first stage we call interphase. Interphase is broken down into 3 separate parts. So when a cell first divides or is born, we would say it begins to grow. It grows and it makes organelles during G1. Now, once the cell gets large enough, it's getting ready to start the process of dividing or becoming 2 new daughter cells. At the end of G1 or gap 1, there is S phase. We also call this the synthesis phase. This is where the cell continues to grow, but something very important happens in that DNA replicates or makes a copy of itself. It is important to point out that whenever cells divide, they all need a complete set of DNA or chromosomes. Following the S phase is the third phase, G2 or gap 2. In this phase, the cell continues to grow and makes final preparations to begin to divide. So we'll talk about mitosis in its own section, which is these stages right here. This is the part where the nucleus of the cell divides. Lastly, in cytokinesis, just a quick preview, is whenever the rest of the cell divides, so the cytoplasm, the organelles, and the membranes break off from each other to form 2 new daughter cells, and then the cycle again repeats. So in this slide, just reviewing the 3 parts of interphase, this is where the cell spends most of its life, and it's broken down into these 3 parts as you see here. The first gap phase, the synthesis phase, and the second gap phase. The first gap is when the cell grows and it makes copies of organelles. The synthesis phase is when DNA replicates, which is an important process that we'll talk about in this section of notes. And then finally, after replication is complete, the cell continues to grow and prepares for division in the second gap phase. So let's quickly overview DNA replication. You'll need to remember the different structures of DNA that we talked about in the first section of video notes. But let's kind of break down some major parts. First, DNA replication, as you guys know, occurs during the synthesis phase of interphase in the cell cycle. It occurs within the nucleus of the cell as that is where DNA is kept safe. We say this is a semi-conservative process. What that means, as you can see in this image right here, you've got 2 DNA strands or a double helix. These are your original strands of DNA. Then they break apart and they are used as a template to make copies off of. So breaking the bonds between the bases to use these blue strands as a template for the new daughter strands of DNA. That is why we say it is semi-conservative. and that whenever the DNA is completely done making copies, each strand of the double helix includes the original parent strand, and then the other strand is the new daughter strand. It is important to point out that in the cell cycle, there are many checkpoints along the way to prevent copying errors, and we'll talk more about those. Those are called mutations. So the first step of DNA replication is strand separation. So before you can make a copy of DNA, you've got to break these bonds between your nitrogen bases. And remember, these are hydrogen bonds between the bases. So cytosine and guanine, each of them have 3 hydrogen bonds, and adenine and thymine each have 2 hydrogen bonds between them. Helicase is an enzyme, remember, a protein with a specific function, and it ends with ase, so that's how you know it's an enzyme. It breaks these hydrogen bonds between the 2 parent strands of DNA almost like a zipper would work. The point where these 2 strands separate from each other is called the replication fork. So you can see at the replication fork, these bases are exposed so that new nucleotides can be added to complement them. That leads us to the second part of DNA replication, base pairing. So DNA polymerase is another enzyme that's really important to this process. And in the nucleus, there are these free-floating nucleotides. So these nucleotides for DNA that include the 5-carbon sugar, the base, and the phosphate group. They get added by this enzyme to complement the exposed parent strands of DNA. So the daughter strand will be added 1 base at a time by the DNA polymerase. So here a thymine would be added, here a guanine would be added, here an adenine would be added, and so on and so forth. And the same process would occur on this parent strand, where an adenine would be added, cytosine, then thymine, and adenine, and guanine. So that's how this process works. And again, DNA polymerase is your proofreading enzyme. So it's also going to check for errors while it's adding nucleotides. This is a very energy-intensive process, so it does require ATP to make the copies of DNA. Remember, the cellular form of energy is always going to be ATP. Step 3 of DNA replication is whenever everything gets kind of sealed together. So a different enzyme is used here called ligase. Ligase will seal the bonds of the newly formed DNA together, so it strengthens the bonds between nucleotides and the bases. This process results in 2 identical copies of DNA, almost like a complete set. Each new DNA molecule, again, is composed of 1 of the parent strands and 1 of the daughter strands. This is why we call it a semi-conservative process.